Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Michael Betts. He's a professor of microbiology at the Penn Institute for Immunology. We'll be talking about immunology and our response to it, and our microbiome's response to it. So, Mike, thanks for coming.
Thank you very much, Richard.
Yeah. Tell me about your research and your work. What do you focus on currently?
Well, currently we're focusing on trying to understand human immune responses to COVID virus. Historically, my lab has been exclusively working on studying the immune response in people to various viral infections, focusing on HIV, but also other common viruses that we all get, including influenza and Epstein-Barr virus, cytomegalovirus, and so on.
How diverse? I know the mechanisms by which viruses infect us are diverse, and I'm sure our response is probably diverse as well. So can you step through some of the viruses that you worked on and talk about, how do they affect us, and then how do we respond? Just some of the highlights.
Well, actually, a lot of the typical viruses, the initial response is pretty similar. So, HIV infection or EBV, which of course causes mononucleosis, influenza, all these things during the acute period of time of infection manifest in very similar ways. They may be doing different things inside your body, but you feel the same way. So you will feel an acute fever, myalgia, which is being tired and listless, and you will essentially go through this phase where your immune system is very, very active, and that's essentially what makes you have these symptoms. And during that time period, you will generate an immune response that either will or will not be able to eliminate that or control that infection. And so in a case of something like influenza, you generate a very strong immune response, usually as a child when you're first exposed or by a vaccine, and then you will clear that virus. And you will generate a long-term immune response to that will protect you in the future. Something like HIV or EBV or CMV, you generate the same type of response at the beginning, but they then manifest in different ways because your immune system is unable to effectively clear those. And so you then are forced to live with them for the rest of your life.
When your immune system is unable to clear a virus, is it the virus adapting and then taking a different tact, or is it your immune system forcing the virus into a new regime where is it continuing its previous action? It's just manifesting differently because of the action of your immune system. Is it possible to figure that out?
It's a really great question, and basically every scenario that you just introduced kind of happens. In different cases. So if you take something like EBV, Epstein-Barr virus, or CMV, cytomegalovirus, the human population has lived with these viruses for tens of millions of years, and those viruses have adapted to live with us without killing us, pretty much, unless you get immunosuppressed, and then they can cause problems. And they evade your immune response in some ways. They use it in some ways. They sequester themselves in places where the immune response may not be as efficient at eliminating them. For instance, something like HIV, which has been in the population, of course, for a much that we know of, for a much decreased amount of time, in the order of maybe 100 years that we know of. That virus is still is adapting continuously, and inside a person who is infected, that virus continually mutates to evade the active immune response against it. And of course, it is pathogenic in many, if not most people, but it takes, of course, a long time. And during that time, the immune response will continually try to be effective, but the virus is always a step ahead in this case. Something like flu, you have sort of different situations of antigenic drift and antigenic shift, which are well documented. It's not the topic of my research, but that's sort of a more population level change in the virus over time, either to evade antibody responses or whatnot.
Based on how viruses change that stay with us, HIV, cytomegalovirus, et cetera, how do you guess that the virus is, it appears that as if it's coordinating its activity, or no? You think it's using, our cells and has some kind of viral quorum sensing or, coordinated action?
Well, unlike bacteria, which can kind of, I think, dynamically respond to their environment, viruses, of course, they're just machines in a sense, and so they respond, but they don't do so proactively. It basically is more of a survival of the fittest situation for viruses. So the best case example would be HIV, where you have this continual outgrowth of mutations, and the reason why that happens is not because the virus knows what's going on. It doesn't have a way to proactively adapt. It's essentially the virus is replicating to such a high degree, and it's the way that during its replication, it makes a lot of mistakes, and probably the vast majority of those mistakes are dead ends for the virus, but some of them will not be, and those that are not then are going to be the viruses that the immune system sees. And some of those will again be dead ends because the immune system will be effective against it. But then there's even one or two at any one point that might not kill the virus itself, and then also allow it to evade the immune response. And that might sound like a really inefficient process, but you have tens of millions of viruses, new viruses produced per day in a person who's actively infected. And just by chance, you're going to have these variants occur. And then they outgrow, and then your immune response is forced to adapt to those again and again and again, and the virus adapts again and again continuously. Again, it's not an active process; it's a passive process in this case for all the viruses. The really long-term viruses that have been in the population, the herpes viruses, including EBV and CMV, they have co-evolved with us for a very long time and have very many actually very interesting components of their machinery, proteins that are either completely homologous or partially homologous to our own proteins. And they do this because they can then modulate to some degree the way the immune system responds to it. So it almost seems like an active thing, but basically they've stolen pieces and they've mutated to allow the virus to persist.
Yeah, what are some of the ways in which CMV and the herpes-related viruses have co-evolved with us? Any specifics that are really interesting? So
probably the easiest one I could think of quickly, and I'm not sure how much you want me to get into the details here. So CMV encodes a homolog to a protein called MHC class 1, and MHC class 1 stands for major histocompatibility complex 1. It is a protein that's found on nearly every human cell. And this is the primary protein that is recognized by CD8 Tcells, which are actually the type of cell that my lab studies. And the function of CD8 Tcells is to kill. One of the main functions is to kill or suppress virally infected cells. And so CMV through, and really I have to say that the mechanism is not super well defined, but CMV encodes this MHC homolog, and it's thought to either be a decoy or to distract the CD8 T cells, or maybe even to render them anergic or non-responsive in some ways. such that the CD8 comes along and sees this protein that normally it uses to target the cell for killing if it's infected, but now it's getting this alternate protein that maybe in some ways allows the infected cell to not be recognized efficiently. There's other proteins that the herpes viruses express, like cytokine homologs. So cytokines are proteins that can be growth factors for the immune system, different types of cells, or even non-immune cells. They can activate the cells in different ways and sort of in some ways potentiate immune responses. And so the herpes viruses also encode homologs of some of these different types of cytokines as well. And again, just sort of probably modulate the immune system in just enough of a different way that it allows the immune response to not be as effective.
So and then, is the virus doing this when it's inside a given cell that normally would be targeted by the immune system and it's masking it? Or is this on the virion itself? It has this protein, so the virion hides.
So the things I'm talking about here, and specifically, would be the infected cell. So when you think about things on the virion itself, that's mostly going to be impacting potentially antibody recognition. And for example, when you think of the flu vaccine, you're primarily stimulating antibodies to allow you to maybe not 100% prevent you from ever getting infected with the virus, but clearing that virus or just decreasing your chance of getting it because you have antibodies there. The things I'm talking about here are more things that are important for T cells, which is what my lab studies. The other really fascinating thing, and actually this actually is really the topic of what our lab has been focused on for the last several years, is the idea that not only do these viruses, and this is HIV is especially doing this, not only do they modulate the immune system, but they actually are taking advantage of the immune system's sort of normal processes to allow themselves to continue replicating. And what I mean by that is they preferentially infect types of cells that are physically located in your body in areas where CD8 T cells normally they're present, but they don't necessarily have the same functional ability to eliminate those cells. So here in this case, we're talking about lymph nodes, and of course, lymph nodes. Whenever you have an acute viral illness, your lymph nodes swell up, and that's when you feel your neck, and you can feel these swollen things in your neck. Those are your lymph nodes in your neck, and that's sort of the site of priming of the immune response. But it's not normally a site where you would want an infection to be happening, if you understand what I mean. But in the case of EBV and HIV, their primary targets of those viruses are cells that reside in those tissues. And so in a sense, it's also another level of evasion that these viruses are doing by taking advantage of being in a place where it almost might sound counterintuitive. The lymph node is a place where you induce immune responses. But it's also a place where you're not thinking that you have a pathogen there. You're not thinking you need to keep an immune response going there. And what we found is that CD8 T cells, which are commonly called killer T cells or cytotoxic T cells, they are present in lymph nodes, but they actually don't seem to kill things in lymph nodes. They do that outside of the lymph nodes. And we believe that this is essentially an evolution process that you don't want to eliminate the cells that are important for making immune responses. That's what would happen if a CD8 was killing things in your lymph nodes. And so these viruses, especially HIV in this case, very effectively hides in these lymph nodes, and therefore you have ineffective abilities to control it.
Well, I know, like in the, I believe it was the 15 or 1600s, René Descartes said that animals were living machines, and then for a while, people thought bacteria were living machines, and now it seems like a growing number of scientists are saying viruses are alive too. So maybe it may be good to consider at times that they are alive, and then look at their behavior in that context and see if that tells you anything different. It's just an idea.
I don't think there's any evidence that there's any brain to a virus, right? It's an exceptionally efficient machine that adapts to its environment, but it's not doing so consciously, or proactively in a sense, right? It just it's all a numbers game with viruses. If you remove 99% of the virus because of an effective immune response and 1% gets out, that's fine for the virus, right? It doesn't care. One of the things we study a lot in the field that I'm in is CD8 T cell escape in the sense of HIV, where the virus is very, very adept at just making single amino acid mutations that completely abrogate the ability of CD8 T cells to recognize an infected cell. But the virus doesn't have any idea what the CD8 T cell is recognizing, right? So we're talking here about, a relatively decent sized virus, and it's a 9 amino acid stretch of protein that the CD8s recognize. So how would the virus know proactively that this 9 amino acid peptide would be what a CD8 T cell targets? It has no idea because it doesn't have ideas. And essentially it's just the numbers game. The virus is mutating continuously without pressure. Without pressure, it still mutates because it just makes a lot of errors. Most of those, as I said before, are going to be dead ends. And then you'll just by chance have that one virus that's mutated the right spot, and it grows out, and then it takes over the population. So. So
you're saying that if it's composed of, let's say, 9 amino acids, one gets modified, and there are cases where that allows it to escape the CD8 cells? Absolutely.
Yeah, there's very beautiful literature on this from when escape was first described that showed HIV, and this is work from Rodney Phillips' lab in Oxford, showed in monozygotic twins who were both HIV infected. So they were responding to they had the same MHC class 1 genes. Their CD8s were recognizing the exact same, in this case, 10 amino acid string, if I remember correctly, and from the entire virus, right? And in one of the twins, the virus figured out how to escape by random chance. And that particular individual went on to progress rapidly and died, whereas the other twin, the virus did not manage to escape, and the CD8s continued to be effective. So just a single, it's not always this black and white. This is a very idealized circumstance, but it just demonstrates the power that these CD8 T cells can have in limiting the viruses, and also tells you the importance, how important they are from the virus standpoint. The virus doesn't like them. And it would want to escape from all of them if it could. Sometimes it manages, in the case of HIV, for the most part, it manages to do so quite efficiently. Things like EBV and CMV, they do not have the same degree of error generation. So they instead, they're not very efficient at all of escaping these responses, but they have these modulatory capacities that I described earlier.
So in a given host, because of the high rate of mutation of viruses, I would think you'd have eventual populations of different kinds of viruses. And I don't know if they would compete or if they would just, infect a person in different ways. Like, what happens over time in someone with HIV or, Epstein-Barr, et cetera? Like, have their viral loads been sampled, and is there a diversity in population because of these mutations?
Yeah, so let's just be very clear. The herpes viruses like EBV do not do this. Okay, so this is specifically talking about HIV in this case. So and the answer to your question is absolutely. And so there's a great degree of evolution of the virus within an individual, and at any one time you'll find what's called a quasi-species within one infected person. There will be typically a dominant virus, which may be sort of the virus of the day that happened to be the most fit variant that just happened to start replicating that day. But then you'll find a whole array of what we would call singletons, just, depending on the depth that you're sequencing. And you'll just find, tons of these more rare variants. And 3 days from now, one of those rare variants might be more fit than the virus that you had 3 days before, and you'll see the swing in the whole population. The other thing that's really interesting about the virus is, and this was demonstrated a number of years ago, actually, by Simon Malal, is population level evolution of the virus as well. And what I mean by population level, I mean the human population. So and this is in regards to CD8 T cells. And so basically what happens is, if one person is infected and they have their particular MHC alleles, which are what CD8 T cells recognize, a peptide from the virus attached to that MHC, one person's infected, and then so they have their set of these MHC genes, and the virus has evolved within that person to be effective at escaping those immune responses. If that person transmits the virus to another person who has either some or all of those same MHC genes, the virus does not need to escape because it's already escaped in the first person. It goes into a host now that also is going to apply the same pressure. And so you will then essentially keep the virus evolving in that same direction. If you transmit to a person who has different MHC alleles, you now force that virus to recalibrate, and it will do so very efficiently. But it will change. And so essentially, if you have the virus going through populations of people where you have a lot of shared MHC alleles, you'll start swinging the virus towards an accumulation of escapes that can deal with that particular MHC allele, if you understand what I mean. So evolution within and amongst people, right?
Yeah, when a virus changes certain cell populations, MHC, so it's literally contributing an extra peptide to those cells, and their MHCs are changed forever, or is there a chance to observe them? Or
no, the host cells are not altered in this sense. No, this is the virus is adapting. The virus is changing its sequence. Yeah. It's not the host cell changing anything. Not in a traditional sense. No.
Well, how is the change in recognition accomplished if it's just the virus itself changing?
So
like where is the recognition event happening in this case?
In this case, MHC, the MHC protein is expressed on the cell surface of an infected cell. But whenever that protein is produced inside the cell, you have a piece of the virus attached to it. It's basically the nature of the MHC to bind to these short peptide fragments that are derived from both host and from any protein that's made by the cell. It can get chopped up and put into this MHC molecule. And that MHC is then transported to the cell surface where CD8 T cells are continuously scanning all over your body, looking for things they don't like, in a sense. And as a neonate and early in life, your body trains itself to not recognize itself very effectively.
Right.
So then it can tell the difference between something foreign and something self. And so when the virus is inside of a cell and it's replicating, it's producing its proteins, those foreign proteins get chopped up along with everything else, and they can get randomly put onto MHC, and that MHC is put to the cell surface. And then the CD8s recognize that peptide from the virus in the context of the MHC molecule, and then it triggers them, and they kill the target through a variety of mechanisms, or they respond to that target, that infected cell.
I get you, but is there ever an event where that cell is now it can somehow clear the virus, and if so, does it retain the MHC modification, or is there ability for that cell to divide, and if so, are the daughter cells containing the MHC variant?
So
ever happen?
Yeah, it's not a variant. Okay, so it's not a permanent change, right?
Alteration.
Well, it's not even an alteration. So the piece of the peptide is not permanently. It does not get encoded into the cell. It's not part of the genome of the cell. That peptide can fall off of the MHC, so it's not a permanent bond. The MHC is just designed to be able to bind to these fragments. It doesn't alter the MHC protein backbone itself whatsoever. So that MHC, which has this piece of the virus stuck onto it, essentially, that's what's getting presented. That has a certain amount of time that it's accessible on the cell surface to the CD8 T cell, and then it goes away or falls apart or whatever.
Okay, so it's like a temporary,
Exactly.
it's like wearing a hat, I guess, like a baseball hat or
That's
something.
that's a very good way to look at it. Yes, yes, and you could change your hat. It doesn't matter, right? Exactly.
Okay, well, that's interesting too. The fact that it's not permanent, that's also useful if it could be harnessed somehow.
Well, that's a basis of there's a lot of vaccine strategies out there to try to stimulate CD8 T cells, and you can in mice it's a very effective strategy. You can just make a plasmid that encodes one peptide sometimes, and if you do it the right way, you can put that into the animal. The cells will start making this one single peptide that's foreign, and you can generate CD8 T cells against it very effectively. Unfortunately, that simple strategy hasn't worked in animals higher up than mice, really, not effectively. But this is essentially the premise for a CD8 T cell based vaccine or therapeutic in this sense. Yes.
When you're amping up CD8 cells to recognize, other things, how do you do that? And is it a training process? Like, do you expose them to, these peptides that would normally alter MHC and therefore they can recognize it? Or, how do you train the cells to recognize things they should or they normally wouldn't?
Well, I mean, to be totally fair, this is not exactly what my lab does. But certainly, in the sense of a vaccine, typically what one would do is you might, well, first of all, just giving a foreign protein or peptide in this case can just generate the CD8s, if you've done it in somewhat the right manner. You can further amplify them by adding cytokines that might make those CD8 T cells proliferate better or make them differentiate into different states. There's a variety of cytokines that can do this. And so that's sort of thinking about like adjuvants that are commonly used in vaccines might sort of change the complexity or the flavor of that immune response and skew things towards CD8s or skew things towards other types of T cells like CD4 T cells, or maybe even skew towards an antibody type of response. Is commonly done in the vaccine field.
So, I've been asking you the questions I want. What are some of the major questions that you're trying to answer right now with your research? And is it in relation to COVID? Like, are you deep in it with COVID yet? Are you?
Oh, yes. Absolutely. Yes, we are. And so, but let me start with the first part of the question. So, my passion for this research is really maybe somewhat self-serving, is to try to understand how we, or even myself, fight these diseases. And I've just always been fascinated by this, and in my particular lab focused on T cell responses as the direction, although there are plenty of different directions one could go with that type of a global question. It's really just to try to understand how people fight these diseases. And so we've typically study, and I should say most immunologists such as myself typically would be doing this from animal models like mice or maybe monkeys. We've tended to bias everything we do towards trying to understand immune responses in people, because that's what I really want to know about. And so of course, COVID presents from the scientific point of view, an absolutely fascinating prospect for studying immune responses in people. And I'm not trying to, this has been an awful thing, of course, scientifically fascinating, from the humanitarian side, awful. But we have really taken it upon ourselves and collaborating with absolute fantastic people at Penn to really try to help define exactly what happens to the immune response during COVID infection. And really this comes from, and it's a field that's only, I guess at this point, 5 months old. We're still as a field learning everything from the bottom up on this. There certainly are expectations. We as I've described at the beginning of our conversation, we know many things about what an acute viral response should look like. COVID, in some senses, is no different than another viral infection from the immune system perspective. The thing that's different is of course, the pathogenesis of this and the wide array of clinical outcomes that seem to happen, and the manifestations of those outcomes are just fascinating. And we're still continuing to try to understand even how the immune response is changing during the acute infection, how it evolves over time. There's questions of whether you generate long-term protective memory, immunity, so if you get challenged again, if you'll be protected or not. I believe that people do so, but there's been some talk that maybe they don't. And what's happened in my lab with this is that we're trying to we took a step back from what we would normally do, and we just really started off with broadly characterizing everything that we could from a given person's blood who had severe disease, and really asked the question, well, what's happening to their immune response during this time? And there's certainly other labs that are working on this too. We have seen some things that other people have seen. We've seen lots of things that other people have not seen in their research. And what's allowed us to do that is the scale of the study that we've been able to do because of the really fantastic work of the clinicians at the University of Pennsylvania and the logistics of setting up the ability to access these patients, that of course the generosity of the patients and being willing to participate in research as well. Cannot understate that, or their families. But really the question that we're trying to sort of start with is, we normally think of the immune response as a good thing, but as I said at the beginning of our talk, the immune response is also what makes you feel sick, right? So the question is the immune response the culprit of the symptoms here, or is it something else? And to be totally honest with you, we don't know the answer to that yet.
Well, what's novel that you've seen about this, coronavirus so far in your experimentation?
Sure. So there's two aspects of things general aspects of things that we've been looking at to do with the immune responses. And so there's two types of immune responses. We've only talked about T cells so far in our conversation, a little bit about antibodies and B cells, but there's a whole other arm of the immune system, which is called the innate immune system. And these are cells mediated, the cellular part of the innate response are mediated by other types of cells that we haven't discussed. And these would include natural killer cells or monocytes or neutrophils and so on. And so probably the most fun thing I've had with this so far, scientifically again, is really seeing the impact that severe COVID-19 has on the innate lymphocyte populations. And this is something that really hasn't been appreciated in a very strong way yet in the literature. And that's basically because a lot of the work that's been done has been just done on a small number of individuals with severe disease. And in our case, we've been able to look at enough individuals to really see what's happening at more of a cohort level. And so what we're seeing is sort of very dramatic changes in the cell surface protein expression of things that are important for these cells to function.
One of the two predominant ways in which, SARS-CoV-2 infects people. I've heard a lot about the ACE2 receptor.
Yes.
That, the case.
Sure.
Talk about that.
Yeah, no. So that's virology and pathogenesis in that sense regarding, host cell entry. This is not, as far as I know right now, this doesn't really have anything to do with that side of things. So in particular, what we're finding, there's some very early things the immune response tries to do in response to any virus infection or any actually infection in general. It doesn't even have to be viruses. And this is the job of the innate immune response. And so one of the major things that we see happen is a large effect on a population of cells called neutrophils. And neutrophils are really something I'm just learning about it in some senses, but they're really, really interesting cells. They're essentially programmed to die very quickly and once they get triggered, and they actually turn over very fast. And what they do is they are very efficient at homing to any area of the body where there's something's gone wrong. And when they get there, They can respond by releasing proteins that will call in other lymphocytes, for example. They can respond, in fact, by dying. And when they die, they actually release their DNA, and in so doing, that also can attract other cells. They form these things called nets, and essentially can clog up holes. There's beautiful videos of this that have been published by Ron Germain that shows neutrophils going into the tissue from the blood to plug a hole that was made by a syringe. For example, they're really interesting. They're suicidal cells, right? But their job is very clear. So what we see happen actually in COVID is an extreme elevation of these cells in the blood. And at the same time that the frequency of these cells elevates, you have changes in the molecules in their cell surface, which are curious, and we don't really quite know what the ramification of this will be. So two of the proteins that we've been able to find that are changed are two molecules called CD15 and CD16. And these proteins have different functions. And just to put it very simply, CD15 is involved in telling those cells where to go. So CD15 will be involved in making the cell have access, for example, to leave the blood to go into lung tissue. Okay, just keeping it COVID related here. The other protein that we see a large effect on is CD16. And CD16 is not necessarily involved in this trafficking type of thing like CD15 is. CD16 actually binds to antibodies. And remember, antibodies are what can bind, in this case, to viruses and allow you to clear those viruses or whatnot. And so CD16 gets affected on the neutrophils as well. And in both cases, these proteins decrease on the cell surface of the neutrophils in the blood. We do not know why that's happening. We do not know what the functional consequence of that is. But it's a very pronounced effect in people with severe disease. When we look in people with mild or moderate disease, we do not see this effect. When we look in people who have recovered from COVID, this effect is gone. It's returned to normal. And when we look in people who are followed longitudinally, so they check in the hospital, we get there. So essentially the way our study works is when the patients first come to the hospital with their severe symptoms, severe enough that they had to go to the hospital. So presumably they're probably been sick for a little while, but now they're starting to really have a lot of trouble. That's our first time point. And that's when we see this effect take place. And if we follow those same people, so let's say a week later, in some people, we see a recovery back to normal for these markers. And in other people, it stays the same. They're still substantially decreased. We're trying
these markers are localized, again, to a particular cell type and an area on a cell, or are they present?
Yeah, so these are cell surface proteins, and they're not found on every cell in your body. It's only certain cells that have these. And for this, I've just been talking about neutrophils, but we see this same effect happening on essentially every cell type that expresses these proteins that are lymphocytes in your blood. So I won't say it's across your whole body because I don't know that. But when we look in the blood and we look at the different types of cells that express these proteins normally, we see this effect happening on all of them. And so what we really want to understand is, can this be used? Clearly, we're only seeing it in the most severely infected people. If we were able to take people who had been recently infected but were not severe yet, would we start to see this change? In other words, would this be a prediction of severity? We hope to maybe determine that, because then it could be a readily accessible way. If a person knew they were positive and they could say, oh, I'm starting to see this effect, maybe I need to be careful. Or if there's a therapeutic that helps to reduce severity, they would know they needed it, for example. This is also
do you have any idea on how long this window is? If it's a 2-week window or 1 week?
So we don't know the answer to that yet. Certainly, the individuals who we've tested so far, which is actually quite a large number, we're probably 40 or 50 people now we've observed this in. They, again, the way the cohort the way the patients are, we don't know exactly when they were infected. So we can only know how long they've had symptoms. And so there's a little bit of a time shift there across the different people, right? So it's not like an animal study where one could infect them on day 0 and then 5 days later we start the experiment. We don't have that timing.
It's one thing you could do if a certain person gets very sick in a family, and you ask, you have to get permission, of course, but if you ask the family members that are in the same household that are not yet sick, if they would volunteer to have their blood work done every day for the next, few weeks, you may be able to catch its inception and passing of it,
Absolutely. Yeah. And that is exactly right. And for us right now at Penn, there's been a lot of talk about trying to do that. We have not yet started to do something like that. And it's mostly logistics, Richard, because if, you have to think about who's going to go and get that blood, who's going to go into the house where there's an infected person and draw blood from the family, right? So there's a danger to the healthcare worker there potentially of exposure. And it's just logistically is complex. It's not impossible. Other places are doing it. It's just something that right now we're still working on the logistics of this and determining whether it will be possible for us to safely do that. But you're absolutely right. But we do have the ability, we do certainly get individuals in the hospital system who do not have a severe disease yet. And we are monitoring them as much as, the ones who get enrolled into the study. There's another study going on at Penn, which is to prospectively monitor healthcare workers, because a lot of them do get exposed, or a fraction of them, I should say, do get exposed. And so they're monitored prospectively. And so there might be a chance to capture some individuals like you suggest in that sense, where we would even have, prior to infection, we would have material to have the perfect baseline. And then, in those, people who do unfortunately do get exposed, then there might be a way to longitudinally follow them as well.
Very interesting. Yeah. I mean, even in more well-characterized issues, like, you mentioned HIV, Epstein-Barr, et cetera. Is there any idea on the window that, the MHC in those circumstances has been masked or, temporarily changed?
Well, okay. Again, remember, the MHC is not changed. It's not.
Well, I'm trying to find the right word for it.
Yeah. Well, essentially you're asking at what point will the CD8 recognize that cell's infected, right? So that's, at what time frame will the MHC be on the cell surface of an infected cell and have that virus peptide stuck to it? Okay. That's essentially what you're asking. So it's actually a very good question. In humans, and there's only a couple of studies that have done this, and again with HIV, again it's a very difficult thing to do. Obviously, you cannot experimentally infect a person with HIV, so you can't track things in an exact way. But there's two groups that have done very, very careful tracking of high risk individuals and follow them prospectively to try to capture exposure events. And in those studies, one run by the Army and one run by the Reagan Institute, both studies were run in different countries in Africa. They were able to get the timing down quite carefully of when the first immune responses appear and when the virus is sort of peaking in severity during that early period of time. And so the virus window, the peak virus is somewhere between 10 to, let's say, 17 days. Day 14 is 2 weeks is typically sort of post-exposure is the highest peak, probably on average. The immune response in terms of CD8s that would be recognizing MHC starts to appear as soon as the virus appears. It can be, and that might be day 7 to 8. You might start, when the virus starts to really pick up its replication, and then you're getting exposure events to the immune system. The day 8 would be very hard to find in people, but in animal models you can find things that early happening, where you can go into more body tissues where the virus is replicating. In humans, you can't do that as well. So we know sort of that time frame. So in COVID, we've also been trying to do T cell responses. The majority of publications, or almost the entirety of publications on this right now, are looking in people who have already recovered from disease. Logistically, it's a little complex to do T cell work on people who are actively infected because of biohazard situations. But we've looked in some people who have recovered, and we're in the process of getting that type of work up. And then once we have a safe protocol to work with people who are actively infected, if there can be a safe protocol, then we'll try to define early T cell responses as well.
Okay. I'm sure a lot of things will be learned that will tie into the initial latency period, when there's no symptoms until someone's symptomatic. So I'm sure a lot will be found there.
Yeah.
Well, very good. Well, Mike, we're just about out of time. I hope I didn't run you around through too many viruses.
No, it's really a pleasure. It's really a pleasure.
Yeah. What's the best way for people to learn more about your efforts?
So we have a website, which is betslab.org. So BETTSLAB.org. We've actually just launched that website. Alternatively, people are welcome to look me up and contact me. I'm happy to answer questions. And we have a Twitter account, which is @betslab. So, yeah.
Okay. Well, very good. Well, Michael, thanks for coming on the podcast. I appreciate it.
Thank you very much for the invitation. It was a pleasure.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.